Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about RNA editing. According to central dogma of molecular biology, genetic information flow from DNA to RNA and from RNA to protein. During this flow of genetic information from DNA to RNA then to protein, there is no alteration in the genetic information. By the process of transcription, the genetic information that is stored in DNA is only transcribed in the form of RNA, but there is no alteration or no change in the genetic information. But the discovery of RNA editing shows that exceptions do occur. RNA editing is an important mechanism that causes change in the information content of gene transcripts. So there are two ways. There are two important ways which can cause RNA editing. The first type of RNA editing can be achieved by changing the structure of individual bases. Such type of RNA editing is very rare. This type of RNA editing results in substitution of one nitrogenous base for another. And this type of RNA editing was discovered in the study of apolipoprotein B gene and in the mRNAs in the rabbit and human beings. So friends, this illustration explains the RNA editing that occur due to the substitution of one nitrogenous base for another. So this is the double-stranded DNA, which is the gene. The code for the protein known as apolipoprotein B. Apolipoprotein B is a lipoprotein that is responsible for transport of various fat molecules in the circulatory system. This is the gene that encodes the apolipoprotein B. So this gene is transcribed in liver as well as in the intestine. In the liver, the gene is transcribed to form ApoB mRNA. This ApoB mRNA is unedited. That means in liver, no RNA editing occurs. So the mRNA is unedited. And this mRNA is translated to produce a apolipoprotein B, which contain 4563 amino acids, whereas the system is different in intestine. In intestine also, the gene is transcribed to form the mRNA, and this mRNA bind with a RNA binding protein that is a deamine, and this is the codon that is CAA. After this, RNA editing occurs with the help of a RNA binding deaminase. The RNA binding deaminase bind with this particular codon and bring about oxidative deamination of cytosine to uracil. So due to this RNA editing, this codon is transformed to a nonsense codon. And this nonsense codon, it terminates the process of translation or protein synthesis. So due to the transformation of CAA codon to UAA codon, translation completely do not take place. This results in the formation of a truncated polypeptide. So this process results in the formation of a incomplete product that contain only 2153 amino acids, whereas the peptide that is synthesized in the liver contain 4563 amino acids. So by this process, the nitrogenous base cytosine is transformed into uracil, and that result in the formation of an incomplete product, that is a truncated peptide chain. There is another pathway also that can bring about RNA editing. The second pathway of RNA editing is more complex. It occurs in trypanosomes. Trypanosomes are responsible for causing sleeping sickness in humans. This type of RNA editing occurs due to the insertion or deletion of uridyl monophosphate, and this causes a major change in the amino acid sequence of the polypeptide chain. Such type of RNA editing is mediated by guide RNAs. Guide RNAs are specific RNAs that are transcribed from distinct mitochondrial genes. So these guide RNAs help in this type of RNA editing that occur due to the insertion or deletion of uridyl monophosphate. How it occurs? The guide RNAs are partially complementary to the pre-mRNAs that result in the formation of a gap. The gap contains adenine. The presence of adenine on the guide RNA in the gap result in the insertion of uracil. This result in RNA editing. So these are the two important mechanisms by which RNA can be edited. What is the reason behind this RNA editing? The answer to this question is purely speculative. So friends, please share and like this video, and also subscribe my YouTube channel, Sam Online Class. See you. Bye bye. Good night.